Hi, it's Miss Bastido, just here to wrap up diversity, which is Unit 3, Topic 1, describing biodiversity and specifically the classification processes. So there's already a first video on this, and that's more about classification of species. This video is going to more focus on classification of ecosystems. And so let's just jump right in. These are the dot points in the syllabus from this section. And so there's things about understanding what ecosystems are made up of. You should be able to classify an ecosystem and then explain how that helps with management of certain ecosystems. And then just being able to describe the process of stratified sampling. So an ecosystem is made up of both biotic and abiotic factors. And in the last video, we talked about what is a biotic versus an abiotic factor. So just as a reminder, abiotic factors are things that are not living. And so that's the soil moisture or the pH, the temperature, the precipitation, dissolved oxygen in the water, wind, all of those things would be abiotic factors. And all of those things do have an effect on the biotic components. The biotic components are the living things. So those are all the different species living in that area. And so an ecosystem is composed of all of these communities of living things, plus the abiotic components. And so a good example of thinking about that areas are oftentimes made up of a varied or wide array of different types of ecosystems. And so if you looked at the Glasshouse Mountains, that there are lots and lots of different ecosystems just within these mountains here. And so we have, eucalypt open forest all the way to a rainforest, and even some shrublands in here. And those are all very different ecosystems, kind of in a small area. So we can classify ecosystems based on their interactions. So the interactions between organisms, we could look at classify ecosystems based on those interactions, or how nutrients cycle in and energy flows in an ecosystem. And then probably what we do most is based on their habitats and microhabitats, we can classify ecosystems. And so a habitat is just a place where an organism lives. So we could think of this habitat as some kind of, closed forest. Maybe it's a rainforest. I can't really tell just looking at it. But this forest here is the habitat of a lot of different species. When we talk about a microhabitat, these guys, I call them pill bugs, but I feel like in Australia you guys call them something else that I don't remember the name of. But these little bugs, they live in this forest, but they have a microhabitat. And so their microhabitat is under rocks and under leaf litter and places where it's very humid and very cool, which typically we'd say probably in this forest it wasn't that cool, but under here it would be cool. And so their microhabitat might actually be a little bit different than the full habitat. And so we have, in Australia, there's some different systems that are used in order to classify ecosystems. We'll just talk about 3 of them, and 2 of them we'll spend a bit more time on than the third one. And so we'll talk about the Holdridge classification system. This is probably the hardest one to use, but I think once you've used it, it gets pretty easy. And then the Specht classification system. And then we'll talk about this classification system here that is used for aquatic habitats or ecosystems. And so this one here is called Holdridge. And Holdridge is based on evapotranspiration ratio, the precipitation in the area, and then the latitude or sometimes the altitude, depending if it's a mountain, that those could be similar, in order to figure out the name of the ecosystem. And so we can kind of practice this. So if I said the potential evapotranspiration ratio of a subtropical forest would be, so I come here and I need to find subtropical forest. Well, here's the subtropical section down here between 18 and 24 degrees. And it's a rainforest. So I need to go over till I get to rainforest, which is way over here. So here's the subtropical rainforest. If it wants to know the evapotranspiration, then I would go up here. So it's everything in here, that's their evapotranspiration. So it'd be less than 0.25. So probably 0 to 0.25 would be the evapotranspiration in a subtropical rainforest. If you use another example, a wet forest with a precipitation of 500 to 1,000 millimeters per year. So if we look over here, we go 500 to 1,000, and it says that we're in a wet forest. So I go down between, everything here is 500 to 1,000. And so there's the wet forest here. And it says, what latitudinal belt is that in? Well, I just need to go over here. It's in the boreal belt. Or if I'm in the altitudinal belt, then it's the subalpine. So if we're on a mountain or something like that, that's the subalpine wet forest. And we'll try one more. What is the name of the biome with 250 to 500 millimeters of precipitation per year? So we go 250 to 500, then we're in somewhere in here, right? And it wants to know the biotemperature is between 6 and 12 degrees. So we know we're in this section, and here is 6 to 12 degrees. So we have to come over, that's cool temperate. We come over between 250 and 500 and cool temperature. That puts us right here at the steppe. And that's the name of that ecosystem. So there's lots of ways that you can be asked questions about this, but hopefully that helped you kind of be able to interpret it a little bit better. The idea is that there are three sort of planes here that or axes that you're looking at to determine different things. In the Specht classification system, we're looking at percent foliage cover. And so Specht classification, the key to using this properly is that we only consider the highest strata or the tallest strata, the tallest trees essentially. And so if we're looking at this transect here, the tallest trees are above 30, right? So we have this tree above 30 and this one above 30. So if I come up here, which is this? Trees above 30. So I'm only going to be using this line here. We just ignore all other trees when we're doing this. And that's probably the biggest mistake students make, is that they'll just look at the percent coverage of all the trees, and then they end up with the wrong percent. So you're not doing the percent coverage of all the trees, just the trees greater than 30 meters. And so if we look here, this tree here is greater than 30 meters, so it's 8.75. We do that by saying this is along a transect, which was 50 meters. So 13 minus 4.25 gives me 8.75. And then I take my other tree that was over 30 meters and I say 35.75 minus 23.5, and that gives me 12.25. I need to add those two together. And so when I add those two together, I get 21. And then I have to define the percent coverage. I have to divide that by the length of the transect. So the length of the transect was 50 meters. So I divided it by 50, multiplied it times 100, and I end up with 42% coverage. If I come back over here, remember, I'm only looking at the trees greater than 30. And I also ignored these two here because they were not greater than 30 meters. And so 42% coverage for talls greater than 30 meters, that would be here, between 30 and 70%. And so this is a tall open forest. And this is sort of another kind of way that you could look at it. If you're looking at it from a bird's eye view, so looking at it from the top, again, we're only looking at the tallest species that exist. Now, occasionally there won't be any trees and there'll be shrubs. So you pay attention you would start down here if there's only shrubs, but these are all trees. So this tree here, the blue one, A, is 25 meters high. So that puts me in this second section here, 10 to 30 meters. So I ignore all these other ones and I only just look at these dark blue ones, and I'm trying to estimate the percent coverage. Now, you could really be specific here and find the area of the circle and then the area of the square, but you can also probably eyeball it and mostly get it right unless it's a really close one, because these percentages here are so big. And so if we did that, I estimate that these blue ones are somewhere between 35 and 40% coverage. And so if we do that, remember that we're in the 10 to 30 because these trees are 25 meters high, and it's 35 to 40% coverage. And so we're going to say that it's an open forest. Hopefully that helps you. The more you do these, the easier they get. So you could get them as if it was a transect, or you could get them like this. You could even get a combination of those two. And then the last type of classification system, this is just used to classify aquatic ecosystems, and it's much more broad. It's not giving a specific name. It's more thinking about levels, as we talk about, like on the regional scale, what's the climate, what is the hydrological cycle like, and then as we get closer into that landscape, what does the landform look like, what does the climate in that specific landscape look like, and then at level 3, we're now looking at, is it surface water or is it subterranean, meaning is it under the ground? And then from there, is it marine water? Is it an estuary? Is it a river? Is it a floodplain? So where is this surface water? What kind of water is it? And then even in the subterranean, is it in the sedimentary rock? Is it in a cave? Is it fractured? So all of these sort of just trying to get it a bit more specific as they go. And then as we get to the habitat, now we're talking about, okay, it might be a marine habitat, but what is the vegetation like in this specific marine habitat? What is the porosity, or what's the water source, especially if it's something like a floodplain or riverine or something like that. And so this is a way of classifying aquatic ecosystems, because they can be quite different from each other, even though obviously, that's an ocean. Like, there can be a lot of differences between different parts of the ocean. And so these will help sort of give us a better classification system for that. And so then maybe we should have started with this, but then why are we classifying ecosystems at all? And so, and how does that help us manage ecosystems? So the 3 examples in the syllabus were about soil health, coral reefs, and old growth forest. So we'll just a quick chat about each of these. But in order to manage ecosystems, if we're talking about soil health, if we haven't classified an ecosystem properly, we might plant the wrong vegetation in the area and manage it incorrectly. And by doing that, we could have a direct impact on the soil health. If we cull animals that are grazing in that area, and the animals' feces is important for maintaining that soil health, then that would be a problem. And so classifying the ecosystem is really important in general for managing species and knowing what the species interactions are in the area, but also understanding how we can manage it better. And so based on, if the soil is supposed to, if we want to increase the soil health, then understanding what are the native plants that grow in the area, as oftentimes those are the best for the soil health. And then what are the native animals that should be in that area, and how can we manage and make sure and keep those animals intact? And so that includes tourism and things too. So how do we manage tourists in those areas if we know that we are trying to increase the soil health? Another example is coral reefs. And so when we look at coral reefs, in order for proper management, they do need to be classified properly. And so classifying these ecosystems is important, especially when we look at like certain coral reefs that we know have been overfished, or so then we can zone them and make sure that there's no fishing in those areas. Or if it's an area where fish are spawning often, we might have some rules set in place for that. We might also have rules set in place. So there's certain catch limits, right, for lots of different fish. And then you could also have, a fishing season for certain species of fish. And so all of these things are true for different species. But if we haven't really classified the ecosystem and understanding where these species are coming from and what their interactions are, then we won't be able to make very good decisions based on managing the fish and when the corals spawn and things like that. So when coral are spawning, there might be some rules during that time period. And so if we haven't classified that ecosystem, we would have a hard time being able to set those rules in place. And then the last one's old growth forest. And in the old growth forest, the same sorts of things would be true. But these are forests. Old growth forests are forests that really have not been disturbed for really long periods of time. So we really want, these are very usually fragmented habitats, and they're usually, and this is true of coral reefs as well, but there are certain species that only live in these sorts of habitats, and so, or these sorts of ecosystems. And so being able to manage these correctly, how do we allow humans in these areas? Do we allow camping, and how is that going to impact the species that are there? There are lots of endangered species in these sorts of ecosystems because they're so fragmented. So being able to manage them and classify them is really important, because if we have two old growth forests that are separated and there's fragmentation, that may be building corridors between those two fragmented old growth forests could be useful and things like that. But if they're not classified properly, we might not be able to do that, or even know to do that. The last dot point here is about stratified sampling. And so we had talked about this previously because sampling has been talked about before, because we were talking about how do we collect data on biodiversity. And so stratified sampling is when we have an area that is stratified, meaning that there is something different that we know about the area that we're trying to sample. And so this is a bit of an example, but if I have this area and this much is a woodland, this much is a cornfield, this much is a soybean field, and this much is a rangeland, there's more woodland than there is of the rest. And so if I just did random sampling, I could randomly end up with the majority of my samples in here. It's not probable, but it could happen. And so if I know that, 25%, we're going to say, maybe it's a bit more than that, of this is woodland, I need to have more samples from this area. And so I think there's 16 on this side and 8 on this side. So we're going to say this is a good portion. We've taken 8 samples from the woodland areas. The cornfield's a bit smaller, so we only took 6 samples. And the soybean field is the same size as the cornfield, so we also took 6 samples. And the rangeland is smaller than all of them, so only 4 samples were taken. Now, within the area, you could still randomly sample. The idea is, let's say, if 75% was woodland, and if 25% was woodland, and 25% was cornfield, and 25% was soybean, and 25% was rangeland, then we would have equal. Then we could just randomly sample, because we could just equally sample each of them. However, if we know, let's say the woodland, not in this picture, but if the woodland was 75% of the area, then 75% of our random quadrats, or whatever you're using for your sampling, would need to be from that area. And so this is you're only doing stratified sampling when there is a difference in strata. So when we do stratified sampling, there are 5 things that we need to do, and these are in the syllabus, so just make sure you're paying attention to this. First of all, we need to know the purpose of our study. Are we looking at how diversity changes as we move away from a river? Are we comparing this site to an undisturbed site? What is the purpose of our investigation? And then we need to choose a site. So we need to select a site that's representative of the whole area. And so, like in this case, obviously this is a small area, maybe from the bigger area, but we've taken a slice of each, the woodland, the cornfield, the soybean field, and the rangeland, to get our samples from. And so selecting a site that really shows the whole area, or represents the whole area, is really important. And then choosing what technique are you going to use. And so here we're using quadrat sampling, assuming these are quadrats, but is a transect more appropriate? And this choice of technique really goes back to purpose. So if our purpose is to compare, let's say, this area with an area that is doesn't have a farm in the middle with a cornfield and a soybean field and a rangeland, maybe we want to so this is a disturbed area, right? And so we could be comparing it with an undisturbed area. And in that case, we're using quadrats. We wouldn't be using transects. If I was trying to see if there's a river on this side and my purpose is to see how something is changing, how the distribution of something is changing away from that river, then we might use a transect or even a belt transect if I wanted to know the abundance of that as well. And so this choice of technique always goes back to purpose. And then what are you doing to minimize bias? And so in this case, what we've done to minimize bias is actually use stratified sampling. And so by using the stratified sampling, noticing that a bigger portion of the area is a woodland, so more of my samples need to come from the woodland area, is minimizing bias. However, if you were to estimate this incorrectly, you could then increase the bias. So if I said the woodland was this much, but then in the end, the woodland was actually about a half of that. Now I've sampled too much in the woodland area. So it's important if you're using stratified sampling, to really understand the difference in strata. And if you don't know the difference in strata, then stratified sampling would not be appropriate. And so other ways to minimize bias is we use random number generators when we're doing simple random sampling. We might minimize bias by making sure that the site selection is random, but then also represents each of the areas. So all of those, and there's lots of other ways that we've talked about previously about minimizing bias. And then lastly, you should think about how would you present your data? Is it a graph showing the Simpson Diversity Index of 2 different sites? Is it showing percent coverage and showing the average percent coverage with standard error bars on your graph between 2 sites? If we're looking at a transect, our data might be showing it as a scatter plot with a line of best fit, or as a line graph showing as we move away from something, because there would be distance on the x-axis. And so thinking about how is that data presented that would be useful in showing what you've learned from the study. And so these 5 things need to be taken into consideration when doing stratified sampling, or really any type of sampling. But you should know the purpose of the study. From that, you should be able to select a site that represents the whole area that you're trying to study. Choose a technique that aligns with your purpose. Minimize your bias. And then know how to present that data. And when you talk about the choice of technique, you should really spend some time thinking about, why did I not choose a transect? Why am I choosing a quadrat here? Or why am I choosing a belt transect rather than a transect? And so we talked about that in another video, but really thinking about your choice of technique. Are you using simple random sampling, or stratified sampling, or systematic sampling? And are you using a transect or quadrats? And then why is that? And why are you not using the other one? I hope you found this video helpful, and Diego and Omar want to know if you have any questions. Just let me know.